So bonjour, my name is Francesc Puig-Castellvi. I'm a postdoc in AgroParisTech, but also in XTA in Ile-de-France. And today I will show the results of my current research, in which I study by using chemometrics the interaction of the microbial community within anaerobic digesters. Let's start first explaining what is an anaerobic digester. An anaerobic digester is a reactor that transforms biomass or sludge into biogas or methane. This transformation is performed by the microbial community that lives inside of the reactors and use the biomass as a source of nutrients. In this study, we wanted to monitor these microbial communities by using the 16S ribosomic RNA sequencing technique. With this technique, we sequence the RNA of the 16S ribosomic units of this microbial community. Moreover, this sequence can also be referred to as OTU, or operational taxonomic unit, and they are specific for each one of the species of the microbia that lives inside of this reactor. Meaning that by analyzing this sequence, we can able to identify these species. Moreover, if we count the different OTUs that we can obtain in the sequencing, we are able to quantify the population of all these species that lives inside of the reactors. So, if we look on this sequencing data, we will see that this data, in fact, is very skewed. This happens because, in fact, inside of the reactor, there are some species, some microbia that dominate over the others. And because of that, for this dominant microbia, they will show a very high OT count of above 3,000, while, on the other hand, for all the rest of remaining species, since they have this small presence, the OT count that we will find is below 5. Moreover, if we intend to analyze more than one sample at the same time, we will find that some species of microbia are found in some reactors and not in others. And because of that, when we will try to arrange the data matrix, we will need to fill the blanks with zeros. And for that, the data is, in fact, very sparse. If we look at in a typical OT data matrix, we will see that for every sample, the number of zeros, it could be around 50% or even higher. So after having introduced how is this kind of data, the aim of the study, in fact, can be divided into two different objectives. The first objective is to establish a econometric pipeline for the analysis of the 16S ribosomic RNA sequencing data, and the second objective is to evaluate the changes in the microbial populations in anaerobic digesters derived from using different substrate composition. To do this, we have used 9 anaerobic digesters, and we put some bacteria, some microbia in these digesters, and then we feed the bacteria with a sludge combined with a co-substrate, either fish waste or vegetal waste. And well, these two co-substrates are rich in organic matter. And also these binary mixtures were performed at different proportions of these substrates. Moreover, we performed this experiment using a time course experimental design, so meaning that we introduced the same inoculum at day 0 in all the digesters, and then we took 2 time points. For the mixture of sludge and fish waste, we took the samples at the day 21st and 28th, and then for the reactors that contain a mixture of sludge and vegetal waste, we collect the samples at day 14th and 21st. We used these 2 different time scales because we observed that the production of methane was different for these 2 different mixtures. So we collected the samples, in total 19 samples. We performed the ribosomic RNA extraction. We sequenced this RNA, and also then, by using bioinformatic tools, we assigned this sequence to OTUs. Then we counted the OTUs, and we built this matrix of 19 samples, 1 per reactor and time point, and 1,145 OTU. Since we were dealing with a very complex experimental design, we decided to split this dataset into 2 different subsets, one for each co-substrate. And we analyzed each one of these 2 submatrices by separate. Also, by doing this, all the reactors that are comprised within each of the subsets has been analyzed on the same time points. So now I will present the econometric pipeline that I have used to analyze this dataset. It consists of 5 different steps. In the first step, we remove the OTU that have been found in less than 2 samples And we did this, we did that just to reduce the sparsity of this data set, and also to avoid that, when we will apply the econometric method, we avoid resolving component descriptive of only a few samples. As a second step, we normalize for every sample the EOT counts to 100% in order to remove the differences obtained in the extraction of the RNA. Then we applied Pareto scaling in order to reduce the skewness of the dataset, and we use afterwards on this pretreated matrix the common component analysis, or also called CCA. Then we analyzed the resulting scores and loadings, and for the loadings we applied a variable selection method consisting on the splot, and finally we interpret these selected variables by using phylogenetic data. So if we look for this further step, we performed this variable filtering, and we observed that after performing this filtering, we kept a different number of variables for each one of these two subsets. For the subset regarding to the fish waste subset, we kept only 484 variables, while for the second subset, the number of kept variables was almost 2 times higher. And because of that, we can conclude that the fish substrate is a less friendly substrate for this microbia, and because of that, these microbia can grow on it. Then I will just give you a reminder of what is common component analysis, or CCA. CCA is an unsupervised method in which the components that are resolved are orthogonals, and also in these components, the variables that have the same dispersion of the observations are grouped together, and also at every resolved common component, it will group together the largest number of variables with the greatest effect on a particular dispersion of the individuals. In other words, it means that it will group the largest number of OTUs, and because of that, it has an important relevance from an ecological point of view. And I want to highlight that this method has not been used before in ecological data of this type. So let's proceed with the CCA. We perform the CCA approach in the two subsets, and we observe that these two CCA analyses only reveal two interesting components. And since we have observed that the score distribution is quite similar for the two subsets, I'm going to proceed to explain the score distribution for the two subsets at the same time. So for the first component, we will see that this component is representative of digesters containing only one source of substrate. So for positive scores, we can see that the scores are related to reactors that contain sludge, that are colored in orange, while in negative scores are related to reactors that contain the co-substrate, either fish waste in blue or vegetal waste in green. Moreover, we can appreciate a small time effect, and this is indicated in the scores plots by using a different color pattern. Plain color for the early 10 points and striped color for the later 10 points. If we look at the second component, we can see that the second component is representative of digesters containing a more homogeneous binary mixture. So the positive scores are related to a mixture of fish and sludge in the left, with vegetal waste and sludge in the right, and then on the other hand, negative scores are related to reactors that contain sludge, fish, or waste, or grass. Moreover, in this second common component, we do appreciate a strong effect in this component for some specific samples, in this case for samples that contain this mixture of sludge with fish, and this decline of the scores indicate a loss of the microbial activity in these reactors. So that has been the explanation, the interpretation of the scores, and now we will proceed to interpret the loadings. But because of a time limitation, I will only explain the loadings for the fish waste subset. So in this case, we applied a variable selection using splots. With these splots, we select those variables that are most correlated and and has the highest covariance to the resolved components. And for CC1, we selected 59 variables, and for CC2, we selected 42 variables. Then we combined these two different sets of selected variables, and we found out that, in fact, we had 71 unique variables. So at this point, we wanted to see if these 71 variables were phylogenetically connected, and to do this, we built a phylogenetic map containing these variables. And in fact, what we observed at these 71 variables can be, in fact, classified into a few phylogenetic classes, such as Clostridia, Bacteroidea, Classiomonadia, Neurolineae, Physis-Fariae, Synergistia, and Metanomicrobia. So at this point, we look again to the loadings, and we thought that we could incorporate this information to the phylogenetic maps, and we did. And this is the result for CC1, and also this is the result for CC2. And also this approach of representing like this, the loadings is novel in this ecological field. So up to this point, we wanted to see if we could find any trend on these phylogenetic maps, and indeed we found. So we saw that the positive scores for CC1, which are colored in red, are mostly found in Classiomonadia, Anaerobic, Physis-Fariae, and Metanomicrobia, while the negative scores negative loadings were found mostly in the Clostridia class. For the second component, we found that the most relevant classes were Classiomonadia and Synergistia, while for the positive scores, while for the negative loadings, again, they were mostly found within the Clostridia phylogenetic class. So, after having highlighted all these classes, now is the time of interpreting the results. So we saw that these anaerobic species were found that they prefer to live in sludge, and in fact, the result was expected because sludge is the natural habitat of this microbia. Moreover, we heard that Physis-Fariae has been found also that prefers sludge because it can ferment the carbohydrates that are found in this sludge. Moreover, because of this fermentation, it releases some organic acid that can be consumed from Classiomonadia. For that, it will prefer also sludge. And then in this degradation, it will release some CO2 that can be used for Metanomicrobia to produce methane. On the other hand, we found that these Clostridia species they prefer to live in fish waste. In fact, most of these Clostridia, they present this saprophytic metabolism, or in other words, they decompose the organic matter such as the one of fish. However, it is important to stress that the Clostridia class is a very diverse, heterogeneous class, and also because of that, it is normal to have found some Clostridia species that prefer to live in sludge. And finally, in the mixture of fish waste and sludge, we found some other species that were not found before, the Synergistia and Classiomonadia, and we are that because we are in this mixture, there is a higher diversity of the nutrients, and because of that, there is a less competence among all these bacteria and microbia, and less dominant bacteria can grow on it. Also, it is important to indicate that usually these two microbia are found together. So, as the conclusions of this work, we can conclude that these 16S ribosomic RNA datasets are very sparse and skewed, and in this approach, we intend to reduce the sparsity by using a variable filtering approach, reducing these variables that contain mainly zero values, and then to reduce the skewness, we use Pareto scaling. Secondly, we use this common component analysis approach to resolve the underlying microbial communities linked to this substrate preference, and then we interpret the loadings by using this phylogenetic representation, allowing to interpret these loadings from to obtain a better ecological interpretation of these loadings. And finally, from these interpretations, we observed that these microorganisms are grouped together within these components because of their preference to the because in fact they were living in this habitat, because they prefer the nutrients that are found in these substrates, and also because in these components they have found some other bacteria that also they prefer to live with them. And well, I want to acknowledge all my teams from IXTEA and from AgroParisTech, and also all of you for your kind attention. Merci beaucoup.
Merci Francesc. Une question? Ah, allez, Anna.
Well, you have studied this, have made this study with some particular kinds of bacteria. Do you think this is all the workflow that you have followed can be like a good general procedure to test the suitability of bacteria whose behavior is not so well known in terms of how well they can digest this kind of organic matter?
Well, in fact, in this field, usually what they do is they use a typical inoculum that contains methanogenic bacteria. And then they don't intend to test different other inoculums. But I think, well, that this, since we saw that these results that we have obtained with these chemometric methods, they are coherent with what we can interpret from that, I think that we should, and we can test other bacteria if we can see differences in these responses.
Bien, bah écoutez, je pense qu'on peut remercier de nouveau Francesc pour cette belle contribution.